Welcome to the deep dive. We are going deep today into the world of protein structure.
Yeah.
And this is the really exciting part.
Yeah,
Not just figuring out what proteins look like, but how we're starting to actually design them ourselves.
totally. Yeah, it's amazing.
It's like we're going from reading the book of life to actually writing new chapters in it.
Absolutely. And it's a field that has the potential to really reshape biology and medicine as we know it.
So, for this deep dive, we have a really fantastic research paper. It's quite a dense paper.
It is. It covers a lot of ground.
But it's sort of our backstage pass to this whole field.
Yeah, think of it as the ultimate guide.
It's got a rather technical name. It's called 2503.13522v1.pdf. But
Right, but don't let that scare you.
don't let that scare you.
It's actually packed with incredible insights.
It takes us from those early days of how scientists were even trying to figure out what proteins looked like,
Yeah, like the pioneering work
all the way to the cutting
to
edge.
the latest and greatest in deep learning
It-
and how that's being applied in the real world.
And that's really what we want to try to do today.
Yeah.
We want to unpack this paper, pull out the most crucial nuggets.
Yeah, distill it down
Yeah.
to the
and
essential,
we understand the key concepts.
the breakthroughs,
Where this field is going.
without getting bogged down in all the technical stuff.
Right, we don't want to lose you in the jargon.
No, absolutely not.
So, before we even get to these powerful computational tools,
Right.
how did scientists even begin to understand what proteins looked like?
Yeah, it's a good question because it used to be a real challenge.
It must have been a completely different landscape.
Oh, completely different world.
Yeah.
For decades, our understanding of protein structures relied primarily on experimental techniques, and it was really a painstaking process.
So what were these techniques? What were people doing back then?
Well, the workhorse, the gold standard was X-ray crystallography.
The X-ray crystal?
This allowed scientists to see the atomic details of a protein. I mean, literally see how the atoms were arranged.
Wow.
And the paper actually reminds us that myoglobin was the first protein structure solved this way,
Oh, wow.
way back in 1958.
So this was really the first time we could actually visualize
Yeah, visualized in 3D.
the complexity of a protein.
The intricate atomic architecture of these molecules.
That's incredible, but I heard that getting those protein crystals,
Right.
it wasn't always easy.
Oh, that's the catch. And it was a major bottleneck for the field.
So you couldn't just take any protein and
No.
stick it under an X-ray.
You need to coax the protein to form these well-ordered crystals,
Right.
which it turns out is a really big challenge for many proteins,
I see.
especially those that are embedded in cell membranes.
That makes sense. So membrane proteins, they're kind of like slippery and flexible, and
Exactly. They're not happy outside of their natural environment.
so they don't want to sit still.
They don't want to sit still in a nice crystal lattice.
For the picture.
Exactly.
So then what happened?
Well, then along came NMR spectroscopy,
NMR.
and this offered an alternative.
Okay.
It allowed us to study proteins in their natural state,
Oh.
dissolved in solution.
So not in a crystal.
No, not locked into that rigid structure.
I see.
And the first protein structure determined by NMR was solved in 1985.
Okay.
So we were now able to see proteins in a more dynamic environment.
Instead of a snapshot, it was more like a video.
Exactly, more like a glimpse into their natural movement and flexibility.
That's pretty amazing.
It was a huge step forward.
But were there drawbacks to NMR as well?
There were. It turns out NMR is most effective for smaller proteins.
Okay.
As proteins get larger and more complex, the signals become very difficult to interpret.
So it's like trying to listen to a single conversation in a very noisy, crowded room.
That's a great analogy. Too much going on.
That's too much going on.
The signals overlap and it's hard to make sense of it all.
So it's great for small proteins.
Yeah,
Not so much for the big guys.
right. And then in more recent times,
Mm-hmm.
cryo-electron microscopy, or cryo-EM, has emerged as a real game changer.
Cryo-EM.
Yeah, this technique allows us to visualize protein structures
Okay.
without the need for crystals.
So no more crystals.
No more crystals.
How does that work?
So they cryognically freeze the proteins,
Okay.
and then they image them with an electron microscope.
So it's like taking a snapshot of them in their natural state.
Exactly, frozen in time.
Interesting.
And this has been particularly revolutionary for studying large protein complexes.
So those really big ones that NMR couldn't handle.
Exactly, the ones that were previously really difficult to study.
So these experimental methods, X-ray crystallography, NMR, cryo-EM,
Yeah,
they gave us those first crucial glimpses into the protein world.
the first snapshots of these amazing molecular machines.
But trying to figure out the structure of every protein in every organism
Right.
using just these techniques, I mean,
It was incredibly time-consuming,
yeah, it must have been incredibly,
expensive,
yeah, and slow and just,
and limited.
yeah.
It was like trying to map the entire world with just a handful of explorers.
That's a great analogy. So that's where computational methods began to play a role.
Exactly, and one of the earliest and still very valuable approaches is homology modeling.
Homology modeling.
Yeah, so the idea here is that Proteins with similar amino acid sequences
Okay,
are likely to have similar 3D structures.
that makes sense.
So if we know the structure of one protein,
Right.
we can use it as a template to predict the structure of a related protein.
I see. So it's like if you have a blueprint for one building
Yeah,
and then you need to build a slightly different building,
right.
you can use that same blueprint and just make some modifications.
Exactly, like using a recipe that, works,
Yeah,
but making a few tweaks to the
I
ingredients.
like that, the recipe analogy.
Yeah, and of course, the accuracy of homology modeling really depends on the quality of the known template structures.
So you need a good starting point.
Yeah, you need a reliable blueprint.
And where do you find these blueprints?
Well, there are databases like the Protein Data Bank, or PDB,
The PDB?
yeah, which houses a vast collection of experimentally determined protein structures.
So it's like a giant library of protein blueprints.
Exactly, a treasure trove of structural information.
So homology modeling relies on finding a close relative.
Yeah, you need a protein with a similar sequence.
So what if you don't have that family connection?
Right, what if your protein is an orphan
Yeah.
with no known close relatives?
What do you do then?
Well, that's where threading comes in,
Threading?
also known as fold recognition.
Okay.
So instead of relying on high sequence similarity,
Right.
threading tries to fit a protein sequence to a library of known structural folds.
Interesting. So it's not looking for a specific protein relative.
No.
It's looking for a general shape.
Exactly. It's asking, which of these existing architectural blueprints does my protein sequence best fit into?
I see. So it's like trying on different outfits
Yeah,
to see which one fits best.
exactly. Even if you've never seen that exact outfit before.
So it's recognizing the overall shape,
Yeah,
even if the details are different.
precisely. The embellishments, the colors, those might vary.
Okay, and then we have molecular dynamics simulations.
Right, molecular dynamics, or MD simulations.
This sounds like getting into the physics of proteins.
It is. It's a very physics-based approach.
So instead of comparing to other proteins,
Right,
you're actually simulating how the protein itself would behave.
exactly. We're simulating the movement of every single atom in the protein over time.
Wow, that's a lot of atoms.
It is. It can be very computationally demanding.
So you need a pretty powerful computer.
You do, especially for large proteins or for long simulations.
And how do you set up one of these simulations?
Well, you start with an initial structure of the protein,
Okay.
and you define its environment.
So like what it's surrounded by.
Exactly, like if it's in water or a salty solution.
Right, like the inside of a cell.
Yeah, and then you let the simulation run.
And the atoms just bounce around.
They do, they interact with each other.
And you see what happens.
And we can see how the protein folds, how it flexes, how it interacts with other molecules.
Wow, so we've gone from painstakingly solving one protein at a time in the lab
Yeah,
to these clever computational methods like
Yeah.
homology modeling, threading, and molecular dynamics.
and those were really important steps forward.
But as the paper highlights,
Yeah.
the field was truly revolutionized by machine learning.
Especially deep learning.
And that's where things really exploded.
It's where we saw this massive leap in accuracy and speed.
With AlphaFold being the real star of the show.
AlphaFold, yeah. It's become a bit of a celebrity in the biology world.
It has, and for very good reason.
Yeah, it's been transformative.
So tell us about AlphaFold.
Well, AlphaFold, and especially its successor, AlphaFold 2,
AlphaFold 2.
represent a monumental leap in the accuracy of protein structure prediction.
So before AlphaFold, we had these other methods,
Yeah.
but they were still limited.
They were often limited by the availability of good templates.
Right, you needed that close relative or that general shape to start with.
Exactly, and they weren't always very accurate, especially for complex proteins.
So AlphaFold comes along and changes everything.
It really does. It cracks the code, so to speak.
So what's the fundamental challenge here?
The challenge is this. You have this linear sequence of amino acids,
Okay, the protein's code.
right, like a string of beads,
Right.
and you need to predict how that string will fold up into this incredibly intricate 3D shape.
It's like trying to predict how a complex origami figure will look just by knowing the sequence of folds.
That's a perfect analogy.
And AlphaFold can do that.
It can, with remarkable accuracy.
So how does it work? What's the secret sauce?
Well, AlphaFold 2 uses some incredibly sophisticated techniques. One of the key innovations is the use of attention mechanisms.
Attention mechanisms.
Yeah, think of it like reading a long document.
Okay.
Your attention naturally focuses on the most relevant words and sentences.
Right, to understand the main idea.
Exactly. And similarly, AlphaFold's attention mechanisms allow it to prioritize the most important interactions within the protein sequence.
So it's not just looking at its immediate neighbors.
No.
It's seeing connections across the entire protein.
It's able to capture those long-range dependencies, which are crucial for accurate folding.
Fascinating. And the paper also mentions a recycling mechanism.
Right? The recycling mechanism is another brilliant idea.
So what is that all about?
So imagine the model making an initial guess at the structure.
Okay.
and then it evaluates its own confidence in that guess.
So it's like it's checking its own work
Exactly. And then it feeds that information back into itself
to refine the
to
prediction.
make a better prediction in the next round.
So it's constantly learning and improving
It's a cyclical process, like continuously refining a draft
until it's just right.
until it achieves a high level of confidence.
That's really clever.
And it's especially important for those really complex proteins.
Right, the ones that have a lot of twists and turns.
Exactly, the ones that were really difficult to predict before.
And there's something else in the paper that I didn't quite understand. Equivariant neural networks.
Right, that sounds very technical.
It does.
But the basic idea is that these are special types of neural networks
Okay.
that are designed to respect the laws of physics.
So it's making sure that the predicted structure is actually physically possible.
Exactly, it's imposing constraints so that the atoms don't end up in impossible positions.
That makes sense. So it's not just a random guess.
No, it's a very informed and physically plausible prediction.
Okay, so AlphaFold was this huge breakthrough.
It really was.
But it wasn't the only player in this deep learning revolution, right?
No, absolutely not.
The paper mentions other notable models,
Yeah, there are a few others that are worth highlighting.
like Rose-Tetafold.
Rose-Tetafold, developed by the Baker Lab at the University of Washington, is another very impressive model.
Okay.
And while it also uses a neural network, it takes a slightly different approach.
So how is it different?
Well, it uses a three-track system.
3 tracks?
Yeah, so it simultaneously processes the amino acid sequence,
Okay.
the predicted distances between pairs of amino acids,
Okay.
and the emerging 3D structure.
So it's like having 3 different experts
Yeah,
looking at the problem from different angles.
exactly. And then combining their insights to make a prediction.
Interesting. And this makes Rose-to-Fold faster and more accessible for certain applications.
It does, and it's also been successfully used in de novo protein design.
De novo design, so that's creating entirely new proteins from scratch.
That's right. It's not just predicting the structure of existing proteins.
Right.
It's actually designing new ones.
That's amazing. And then there's OmegaFold.
OmegaFold, yes, that's another very interesting model.
And what makes OmegaFold unique?
Well, OmegaFold was designed to predict protein structures without heavily relying on multiple sequence alignments or MSAs.
MSAs?
Yeah, so imagine you have the protein sequence you're interested in,
Okay.
and you want to find similar sequences from other organisms. Regions of the sequence that are the same across many species
Right.
are likely to be important for the protein's structure and function.
So they've been conserved throughout evolution.
Exactly. They're like the essential parts of the blueprint,
I see.
and multiple sequence alignments are like putting these related sequences side by side
Right.
to highlight these conserved regions.
To see the patterns.
Exactly. And this gives valuable information to the prediction models.
So it's like having a bunch of different blueprints.
Yeah.
From different but related buildings.
Exactly. And you can see which parts are the same and which parts are different.
And OmegaFold can make predictions without relying on this information.
It can, which is particularly useful for orphan proteins.
Orphan proteins.
The ones that don't have many known relatives.
So it's like trying to figure out the blueprint of a building
Yeah.
without having any other blueprints to compare it to.
Exactly. It's a much tougher challenge.
But OmegaFold can still make a prediction.
It can, and it can give us valuable insights.
So we have AlphaFold, Rose-to-Fold, OmegaFold.
Yeah.
Each of these models has its own strengths and ways of tackling this incredibly complex problem.
And they're all pushing the boundaries of what's possible.
Now, how are researchers actually using these tools?
Right. How does this translate into practical application?
Yeah, what are the real-world impacts?
Well, the paper outlines a general workflow for using AlphaFold 2.
Okay.
So you start with preparing the input data.
What kind of data is that?
Simply the amino acid sequence of the protein.
Okay, so you just feed it the sequence.
Yeah, that's all it needs.
And then what?
Then you run the AlphaFold 2 program,
Okay.
which does all the heavy lifting.
All the complex calculations.
Exactly. And finally, you analyze the output.
And what does the output look like?
Often it's a visual representation of the predicted 3D structure.
So you can actually see what the protein looks like.
You can. It's really amazing.
But once you have that predicted structure, how do you know how much you can trust it?
That's a very important question.
Is it just a guess?
No, not at all.
So how do you assess the reliability?
Well, that's where the confidence scores come in.
Confidence score?
AlphaFold 2 provides these scores on a per amino acid basis.
Okay.
They're called PLDDT scores.
PLDDT.
Yeah, and they range from 0 to 100.
So the higher the score?
The higher the confidence in the prediction.
So a score of 100 means?
It means AlphaFold is very sure about the position of that amino acid.
And a score of 0 means?
It means it's much less certain.
So it's like a weather forecast for different parts of the protein.
That's a great analogy. Some areas are sunny and clear,
High confidence.
while others are cloudy and uncertain.
And the paper also mentions the pairwise aligned error? or PAE
The
matrix?
PAE matrix, yes.
What is that all about?
Think of it as a map,
A map.
showing how confident the model is about the relative positions of different parts of the protein.
So it's not just about the individual amino acids,
No,
it's about how they're arranged in space relative to each other.
exactly, especially those that are far apart in the sequence.
So if two regions have a low PAE value,
It means AlphaFold is very confident about their relative positions.
okay, so we have the predicted structure and these measures of confidence.
Yeah.
How does this translate into real-world impact?
So how are these tools actually being used?
Can you give us some concrete examples?
Absolutely. So let's take the example of a single protein like lysozyme.
Lysozyme.
Yeah, you could predict its structure using AlphaFold,
Okay,
and then look at the PLDDT scores for different parts of the protein.
right, and you might see that the active site,
The region where the protein does its job
exactly.
is predicted with very high confidence.
So that's a good sign.
It is. It means we can be pretty sure about that part of the structure.
And what about other parts of the protein?
Well, you might find that some of the loop regions on the surface have lower scores.
Loop regions?
Yeah, these are parts of the protein that are often more flexible.
Okay, so they're floppier and harder to pin down.
Exactly, and that makes sense biologically because they're not as constrained.
So the confidence scores give you a sense of which parts of the structure are more reliable.
They do, they're incredibly valuable.
And what about predicting how proteins interact with each other?
Ah, yes, protein interactions.
Like how an antibody binds to a virus?
Exactly, it's a crucial area of research.
And can AlphaFold predict these interactions?
It can, and tools like Rostitifold are particularly good at this.
So you can input the sequences of both proteins?
Right, the antibody and the viral protein.
And the model will predict how they fit together.
It will in 3D space.
Wow, so we can actually visualize these molecular interactions.
We can. It's like seeing the machinery of life in action.
That's amazing. And what about those orphan proteins?
Right, the ones with no known relatives.
Can we predict their structures?
We can, and that's where OmegaFold really shines.
Because it doesn't rely on multiple sequence alignments?
Exactly. It can make predictions even when there's not much evolutionary information available.
So these prediction tools are really powerful.
They are. They're being used across biology.
From fundamental discovery to drug development.
And beyond.
And this power to understand natural proteins leads us to the exciting field of protein design.
Yes, moving from understanding to engineering.
To creating our own proteins with specific functions.
It's like becoming architects of the molecular world.
That's a great way to put it. So tell us about protein design.
Well, protein design takes the knowledge we've gained from structure prediction and goes a step further.
Okay.
It aims to create proteins with entirely new properties.
So we're not just copying what nature has already done.
No, we're designing proteins that have never existed before.
To solve specific problems.
Exactly, to address unmet needs.
So what are the steps involved in protein design?
Well, the paper outlines a few key steps.
Okay.
First, you need to define the target function.
So what do you want this protein to do?
Exactly. What problem are you trying to solve?
Do you want it to catalyze a reaction?
Right, or bind to a specific target.
Or maybe glow in the dark?
I mean, the possibilities are endless.
That's incredible. So once you have the target function,
Right.
what's next?
Then comes sequence generation.
Sequence generation.
Figuring out the exact sequence of amino acids that will fold into a structure capable of performing that function.
So you need to design the blueprint.
Exactly.
And how do you do that?
Well, there are a few different approaches.
Okay.
One is homology-based design,
Homology-based design.
which involves taking an existing protein with a similar function
Right.
and making strategic changes to its sequence.
So you're modifying an existing blueprint.
Yeah, and tools like AlphaFold 2 and RosDevaFold are essential for this.
Because they can predict how those modifications will affect the structure.
Exactly. You need to make sure that your changes don't disrupt the overall fold.
And then there's de novo design.
De novo design?
Which is creating a brand new blueprint from scratch.
That's right. It's much more challenging.
But also much more powerful.
Absolutely, because you're not limited by what nature's already come up with.
So you can design proteins with entirely new capabilities.
That's the goal.
That's incredible. What are some of the key things you need to consider when designing a protein?
Well, stability is crucial.
Stability.
You need to make sure that your designed protein will hold its shape under physiological conditions.
So it won't just fall apart.
Right. It needs to be robust.
And then functionality.
Functionality, of course, it needs to be able to do what you designed it to do.
Right. It needs to be effective.
And then there are practical considerations like how easy it will be to produce and purify.
So a beautifully designed protein on a computer screen isn't very useful.
Not if you can't actually make it.
So what are some strategies for improving stability?
Well, the paper mentions a few.
Okay.
Carefully designing the hydrophobic core,
The hydrophobic core.
which is the interior of the protein,
Right.
managing surface charges,
Surface charges.
and even introducing disulfide bonds.
Disulfide bonds, those are like molecular staples.
Exactly. They help hold the protein together.
And the paper also mentions optimizing the DNA sequence for efficient production.
Right, codon optimization,
Codon optimization.
which involves choosing the right codons.
Codons are like the words in the genetic code.
Exactly, and some codons are better recognized by certain organisms.
So you need to speak the language of the cell.
That's a good way to put it.
And adding purification tags.
Purification tags are like handles that allow you to easily grab the protein.
I see. So you're making it easier to isolate and purify.
Exactly, but all of this computational design is just the first step.
So what happens next?
The paper emphasizes the absolute necessity of experimental validation.
So you've designed this amazing protein on your computer.
Right.
Now you have to actually make it.
And test it.
So how do you do that?
Well, you synthesize the gene for your designed protein
Okay.
and then express it in a host organism.
So you're putting the blueprint into a cell and letting the cell build it.
Exactly. Common host organisms are bacteria like E. coli,
E. coli.
yeast, or even mammalian cells.
So you're turning the cell into a protein factory.
In a way, yes,
And then you purify the protein.
to isolate it from all the other stuff in the cell.
And once you have the purified protein,
Then you can test it.
so if you designed an enzyme,
Right.
you'd measure how well it catalyzes the intended reaction.
Exactly. You do an enzyme assay.
And if you designed a binder,
You'd measure how strongly it interacts with its target.
so it's a very iterative process.
It is design, build, test, learn.
And the paper mentions some of the key computational tools used in protein design,
Yes, there are a lot of great tools out there.
like Rosetta Design.
Rosetta Design is very popular for optimizing protein sequences.
Okay.
It helps you evaluate different mutations.
Mutations are like changes to the blueprint.
Right, and Rosetta can help you find mutations that improve stability or functionality.
So it's like a virtual laboratory for protein engineering.
It is. It's very powerful.
And then there's protein MPNN.
Protein MPNN, yeah, that's a neural network model that's particularly good at designing sequences that will fold into a specific backbone shape.
So you give it the shape and it finds the sequence.
It's like reverse engineering the protein.
That's amazing. And for visualizing protein structures.
There are tools like PyMol, which
Pymol.
is very widely used.
And even Fold It.
Fold it, that's the game.
The game where you can fold proteins yourself.
Exactly, it's a fun way to learn about protein folding.
So we've seen this incredible progress with AlphaFold and these design tools.
And the field is moving incredibly fast.
The paper mentions AlphaFold 3.
Yeah, AlphaFold 3.
What's the next big leap?
Well, AlphaFold 3 is a significant advancement over AlphaFold 2.
Okay.
The key breakthrough is its expanded capability to predict the structures of a much wider range of biomolecules.
So not just proteins.
No, not just protein.
What else can it handle?
It can now handle nucleic acids like DNA and RNA.
DNA and RNA, the other building blocks of life.
Exactly. And it can handle small molecules
Small molecules like drugs.
like drugs, metabolites, all sorts of things.
Wow, so it's not just about proteins in isolation anymore.
No, it's about understanding how all these different molecules interact with each other.
It's like moving from understanding individual words to understanding entire sentences.
That's a great analogy. It's about seeing the bigger picture.
And how does it handle these different types of molecules?
Well, it's been trained on a massive data set that includes information about all these different molecules.
So it's learned the rules of the game.
It has. It's learned the chemical rules and the spatial constraints that govern how these molecules interact.
And this allows it to make much more accurate predictions.
It does, especially for complex systems like protein interactions and protein-ligand interaction.
Protein-ligand interactions, that's how drugs bind to proteins.
Exactly. And AlphaFold 3 is much better at predicting these interactions.
Which is crucial for drug development.
Absolutely. And it also relies less on multiple sequence alignments.
So it's even more powerful for orphan proteins.
Exactly. And it's more accurate in predicting the structures of flexible and disordered regions of proteins.
Those are the parts that are always whittling around.
Right. They're very difficult to predict.
And AlphaFold 3 is also more computationally efficient.
It is. It's faster and more scalable.
So it's faster, more accurate, and more versatile.
It's a remarkable achievement.
How did they achieve these impressive improvements?
Well, one major innovation is the use of a generative diffusion approach.
Generative diffusion, what is that?
It's a bit like starting with a blurry image
Okay,
and then progressively sharpening it.
until you have a clear picture.
Exactly. During training, the model learns to reverse this process,
So it learns to go from blurry to clear.
which allows it to predict multiple different conformations of a molecule.
Conformation, so different shapes that the molecule can adopt.
Exactly, because molecules are not static.
Right, they're constantly moving and flexing.
And AlphaFold 3 can capture this dynamism.
That's really impressive, and they've also replaced the evil former module from AlphaFold 2 with a new module called Pairformer.
The Pairformer module, yeah.
What does that do?
It further enhances the model's ability to understand the relationships between different parts.